Now that we've looked at transcription and translation, we want to take a look at how gene expression is controlled. First off, let's look at the idea of how does regulation of transcription and translation, the production of proteins, benefit organisms. Every cell within an organism contains a full genome, but not every cell needs to express that full genome. So this regulation can allow some cells to express certain genes and other cells to not express them. And that's obviously very important for a multicellular organism, but even for single-celled organisms, they don't need to express all genes all the time. Even if one cell will ultimately, at some point, express all those genes, we can control when those genes are expressed, and we can control when those proteins are made. And that can allow that organism then to be more responsive to its environment. Regulation makes an organism more efficient. They're more efficient in terms of energy. Expressing all your genes all the time would require a huge amount of energy. We didn't talk a lot about the energy requirement in some of those stages, but remember when we were building that polypeptide chain, every time we created a peptide bond, we had to have energy from GTP to do that. And other steps in this process also require energy. It also allows us to keep cells to a manageable size so that we're not constantly making product that we don't even need. It also benefits organisms because it allows them to express the genes they need more quickly if they're not expressing all of their genes at one time. We know that in multicellular organisms, different genes are going to be expressed in different cell types. And it's the expression of those genes that makes one cell different from another. It's not the genetic content that's there because they all have the same genome. This differential gene expression that gives rise to different cells. Cells of your liver, your pancreas, skin. All those different cells are expressing different genes, have different expression patterns. Now, if we think about prokaryotic organisms, remember prokaryotes have the machinery for transcribing the DNA into messenger RNA and then the translation of that into a protein all in the same area. So those happen simultaneously. In prokaryotic cells, most of the control and regulation occurs at the transcription level. That's not true for eukaryotic cells. In eukaryotic cells, we have a lot of different steps here. There's so many more steps in eukaryotic cells than there are in prokaryotic cells. We can control each one of those steps. There's many points along this pathway in eukaryotic cells where we have points of regulation. Some of the differences that we see in gene expression between prokaryotic and eukaryotic cells has to do with the differences in their architecture. Prokaryotic cells, of course, lack a nucleus, and so we don't separate transcription and translation. In those cells, then most of our regulation will happen at that transcription level. In eukaryotic cells, where we have a nucleus, we have that transcription and translation separated, and we also have that transport of our transcript from the nucleus out into the cytoplasm, giving us another means of control. So we have a lot more control points here. Also remember the packaging of the DNA is different. Because of the way that DNA is packaged in eukaryotic organisms, we have a control point there. We can give it access to the chromosome or not based on how it's wound around histones. We also can regulate transcription like we do in prokaryotic cells. We can regulate the modification after transcription. Remember, we're going to take our transcript, our pre-messenger RNA, and we're going to modify it, add a cap, add a tail, splice it, and that gives us a point of control. Then we also are going to take that messenger RNA and shuttle it out of the nucleus into the cytoplasm. We have a way to control that. Then when we get it to the cytoplasm and hook it into a ribosome, we're going to translate that protein. We can control translation. And then we're going to modify many of our proteins after translation, and we can control that. So there's many more control points in eukaryotic cells that we don't really find in prokaryotic cells. So as we talk about this gene regulation, we'll be looking at prokaryotic and eukaryotic cells separately.